Nice to see you all this afternoon. Can you hear me right now? I can hear myself, so I think it's probably working. So I'm delighted to have been invited to this and to share the responsibility with Louis and Steve. We've talked before this event, and we're going to do our best to give you not only our respective scientific summaries, but also a discussion that Steve will lead about how we put teams together, how we try to get a big problem solved in applied biology. And that's certainly something that I've enjoyed tremendously. I will go through the overview of where we stand today on Alzheimer's treatment from my perspective. It's an incredibly complex disease with a lot of different factors and many more questions that we don't have answers to than that we do. Nonetheless, as a neurologist by training, I'm now prescribing medicines in the clinic to patients, and I've got quite a lot of experience with that, sort of hands-on. We have a group at Mass General Brigham that works together in what we call the ATP, or Alzheimer Treatment Program, and we've treated maybe about 150 or 160, 170 patients with multiple infusions of Leqembi, which is the first fully FDA-approved drug, so I'm going to talk about that a bit. But before that, I'll tell you a little bit about some of the molecular mechanisms, because all of us in this room begin with an intense interest in molecular biology, genetics, protein chemistry, and then its translation. And so my title you can see here, and I have a disclosure at the bottom that mentions that I'm a founding director of Prothena Biosciences, but a long time ago, and Steve was reminding me earlier how long it has been when I was a junior faculty member at Harvard. It goes on forever. You have to have good longevity and persistence. I began a company called Athena Neurosciences, which is now called Prothena, play on words, and they had a lot to do with the development of immunotherapy for Alzheimer's, the work of Dale Schenk, who was a close colleague of mine before he sadly died of pancreatic cancer a few years ago. So the trajectory of what I'll share with you today relates to this headline in our global forum for Alzheimerology, AlzForum, and for other diseases as well. The headline says, "Finally, big win on outcomes for lecanemab in phase 3 top-line results." And a little later on, I'll show you a slide that indicates how many different things have been tried that didn't work. And of course, this has given the whole idea of amyloid lowering a bit of a bad reputation, although I think it's not actually deserved, because so many of those earlier trials that didn't work didn't actually prove that they had lowered amyloid. They were unable to do that. Much of it was before they even had amyloid PET scans. So there are caveats to the headline that I just shared with you, and there's important stuff to be done, as all of you in this room know. So it's good news that we now have a fully FDA-approved therapeutic, but its clinical meaningfulness for patients and families needs to be confirmed over time. So there's been lots of public debate about whether this really moves the needle. I think it does, but it's a question how well. The risk-benefit ratio of lecanemab appears acceptable for many Alzheimer patients, but it's not been confirmed in some subpopulations. And of course, we know that so many of the trials in our field are done with great asymmetry, using people who volunteer, who are very often not typical of our population. So there's a lot we don't know about how well this agent will work, even between women and men, and certainly in a variety of underserved populations. Safety monitoring is very important, because we know that there are headlines in public newspapers about bad events that occur. I'll tell you now that some of the worst events that have occurred with amyloid-lowering monoclonal antibodies, like Leqembi and donanemab and Aduhelm or aducanumab before it, have come because our management of the patients was not ideal. That is, we doctors have to take some responsibility. We've now learned that you do not anticoagulate someone who is on one of these amyloid-lowering agents. You do not give them TPA, tissue plasminogen activator, clot buster, and there have been deaths as a result of that. So as we get more sophisticated and are more careful, especially about ApoE4 homozygotes, which I know Louay will talk about also today, that's where we can avoid some of the bad outcomes that we've seen already. The patient burden is still great. This is an expensive therapy, and it means every 2 weeks you go for an IV infusion. And this is not a zero-sum game, so there are new approaches that many of you may be working on or interested in, and that doesn't need to undermine what we've achieved so far. So it's not we're against Leqembi or donanemab or we're in favor of something else. It's all of these things. Now, this is the kind of graph that got us to where we are now with the amyloid hypothesis. And that is that there is a steady rise in the prevalence of Alzheimer's disease, as you see here. And the presence of plaques in the brain has also been quantified in a number of studies, just a few of which are mentioned here. And it parallels that curve of prevalence of clinical disease, but comes a little bit earlier, actually, about 15 years earlier. And PET amyloid, which arrived around 2002, can show that prevalence of amyloid plaques in living patients. So we don't have to wait till autopsy, we can see it happening now. And that gives us a 15-year interval, approximately, 10 to 20 years maybe, that patients have pathology but don't yet have clinical symptoms. And so more and more the field is defining, although this is controversial, that Alzheimer's begins with the pathology in the brain, and that we can already call it Alzheimer's then, although some people object to that and say, no, you should only use the term Alzheimer's when people have memory and cognitive symptoms. So for years, decades, I've been putting together cascade diagrams of this that are imperfect and not really so linear in biology. John Hardy, my colleague at UCL, has been doing this also, and a few of us, starting with George Glenner, who isolated the amyloid-beta protein, have been behind this idea that amyloid lowering would ultimately show some effect in slowing symptoms. And I'll review with you a few of the key points that have supported this therapeutic maneuver. Alzheimer patients undergo progressive A-beta deposition with surrounding neuritic and glial pathology, and let me go over here, it's easy to read, and in brain regions serving cognition. Down syndrome, as you know, shows typical progression of Alzheimer's lesions, usually already beginning in teenage years, before patients have new cognitive impairment, and that can be caused by gene dosage of wild-type APP. Autosomal dominant Alzheimer's is caused by mutations in the substrate, APP, or the protease presenilin of the biochemical reaction that generates A-beta throughout life. So that's a terribly important point, and this came out of genetics with lack of bias when these genes were discovered, and it turns out they have a direct biochemical relationship. An APP mutation that decreases A-beta production by about 30% lifelong, the so-called Icelandic mutation that Decode Genetics discovered, is a very powerful point of evidence, because those patients don't get Alzheimer's disease. They get very little amyloid buildup even when they're in their 90s and 100. ApoE4 is the major genetic risk factor for late onset Alzheimer's disease, and it decreases A-beta clearance lifelong, and I suspect that Louay will have more to say about its mechanism. Diffusible A-beta oligomers isolated from patients' brains induce neuritic dystrophy and hyperphosphorylation of tau in rodent hippocampus and experimental systems, and iPSC-derived human neurons, and in vivo injection of A-beta from Alzheimer's patients can impair memory. So this is sort of a reductionist approach to take material from the human brain and show what it does to animal models. Human biomarker studies have been very powerful in the last 2 decades, showing falling CSF A-beta precedes rising CSF tau. and then decreased fluorodeoxyglucose PET, MRI, volume loss, tau neuronal spread. So all of these things in biomarker studies have had an orderly progression, that the first element we can detect is the CSA-beta decrease, which means it's becoming insoluble in the brain. And now human trial data show that A-beta antibodies clear plaques and decline and slow decline in cognitive and importantly in activities of daily living. One of the debates again in the field has been, what do you measure to show that the agents work? And the CDR sum of boxes has been widely touted, but the most interesting thing is the ADL scales, and there's as much as a 40% on average benefit for decline. You still decline, but 40% less than placebo. And there's some recent data that it can be much less than that if you start early enough in symptomatic Alzheimer's. So just to review the molecular biology behind this set of observations, APP is a single transmembrane protein spanning the membrane, very simple cartoon, undergoes beta secretase cleavage, releasing the APPS beta ectodomain fragment, and a second cleavage, which is very curious, in the membrane is by gamma secretase, releasing the A-beta monomer. And everything I've shown you in this cartoon is normal, and everyone does this throughout life, and lower mammals, other animals as well. And then when it dimerizes, which can already be detected in some human samples and in some animal models long before behavioral symptoms, these dimers are probably now abnormal. So this is where we shift from normalcy to abnormalcy, although small numbers of dimers might occur all the time throughout life. And the A-beta peptide can trimerize, tetramerize, form dodecamers, and collectively we would refer to these as oligomers, which then, some of which can form protofilaments or protofibrils and amyloid fibrils of 8 nanometer. You can see them under the electron microscope, and you get amyloid plaques. And this is a typical image. This happens to be from the amygdala of a patient that I followed during life, who was 69 years old when he developed symptoms. His mother had died when she was in her early 70s of symptoms. In retrospect, they probably both carried an ApoE4 allele. We didn't know that at this time. And you see 2 round spherical deposits in the orange color with the Bielszowski stain, the very stain that Alzheimer used, surrounded by dystrophic axons and dendrites. So these are neuritic plaques in the center. And then they're lying in a field of neurofibrillary tangles, the dark staining neurons. So in 1999, Mike Wolf, working with me and others at the Brigham, discovered that the enzyme that creates A-beta is none other than presenilin, which Peter St. George Hislop had first isolated and cloned in the Sherrington paper in 1995. Didn't know how it caused Alzheimer's, but it turns out that missense mutations in presenilin 1 and presenilin 2, its homolog, will directly produce a very bad form of Alzheimer's. The youngest case on record is 18 years old. And we identified and studied 2 and only 2 aspartate residues in the transmembrane domain 6 and 7 of presenilin, and we recognized these and thought, aha, could this be an unusual aspartyl protease that generates A-beta? So this was an unpopular idea when it came out, but we did mutagenesis and showed that just mutating one of those aspartates to a glutamate, conserving the charge, or to an alanine, would stop a cell from producing A-beta, and would be lethal in vivo. So this presenilin molecule is required to have 2 aspartates in TM6 and 7 that coordinate with a cis-Al bond and cleave a variety of substrates, maybe more than 100 substrates. By cartoon, this is what we were saying in this paper, that the 2 Ds in TM6 and 7 can access a helically wound substrate that probably needs to slightly unwind helically, as we have evidence for in molecular modeling and in experimental data, to allow cleavage to occur and the release in the purple fragment here of the, of the A-beta peptide. So presenilin is gamma secretase together with 3 other polytopic proteins that are necessary for this reaction. Now, that's about A-beta production. Let's quickly review what the consequences of that are. And the field has gravitated to the notion that the endogenous A-beta monomer that everyone makes throughout life and all animals make and cells make normally isn't really the issue. It's the oligomerization or dimerization. And here you see on a Western blot on the right an Alzheimer brain saline extract with dimers labeled at 8 kilodalton. An identically prepared extract from control brain of that age doesn't reveal noticeable amounts by Western blot of A-beta monomers and dimers that have accumulated. And we then took this process and did size exclusion chromatography, the work of Jin et al., and showed that these dimers, but not the monomers, have substantial neurological and neuropathological effects. So here you see beautiful images of primary rat neurons. We've done this also with iPSC-derived human neurons. And in everything but the last column, you can see that neurons look healthy when you apply control tris-buffered saline extracts. Fractions 10 and 11 are the monomer-rich fraction, but again, this is from control brain. This fraction 8 and 11 with the dimer-rich. And then on the right, it's the dimer-rich fraction from Alzheimer tris-buffered saline extracts that at tiny amounts, less than 2 nanomolar, will dramatically alter the morphology of neurons and cause neuritic dystrophy. And we can rescue this with A-beta specific antibodies. So the monomers in this experiment, in this column, are a terrific control because they come from a postmortem brain from a patient who died of Alzheimer's disease, everything done identically and at the same time. And the monomer fraction, fractions 10 and 11, doesn't have activity on these otherwise healthy neurons, and the dimer-rich fraction does. And those dimers are in equilibrium with larger oligomers as well. Now, the application of these human brain isolates, aqueous saline extracts, to neurons that express tau normally leads to a hyperphosphorylation of tau, a phospho-tau signal. The AT8 antibody, which does this at residues 202, 205, but many other tau antibodies will show increased phosphorylation in the presence of oligomer-rich A-beta extracts. And if you immunodeplete in lane 6 here, the A-beta from these extracts and still have everything else present in the simple saline extract, then you don't get this increased tau phosphorylation. If you eliminate tau, or not eliminate it, but lower tau by knocking it down, you can see that the tau staining in the right upper corner there is decreased. And now when you apply the A-beta dimer oligomer-rich fractions from patients' brains, there's no longer the neuritic dystrophy that you see here. This is a control RNAi. These are two different RNAis that knock down tau. Knocking down tau seems to, in considerable part, protect a neuron from the impairment of OA-beta or oligomeric A-beta. So we think that tau is very much necessary, and it's not coincidence that Alzheimer in 1906 described the presence of both types of lesions in virtually all the Alzheimer patients that he came across. And you need the tau expression to allow oligomeric A-beta to do its dirty work, or at least in part. There are many other features. And then that can be rescued by some, but not all, monoclonal antibodies. So 3D6, which is the forerunner of bapinuzumab, and 82E1, the commercial antibody, but not 21F12, a C-terminal A-beta42 antibody, will, when put onto the neurons together with the oligomer-rich material from an Alzheimer's saline extract, will negate the injury. So this is quite specific. Now, how does this preclinical work take us towards modifying treatment and the prevention of Alzheimer's? And this is where, biotechnology, biopharma has been so important, the areas that you work in all the time. This has been a very long road, and we now have translated this as a field with a great many participants into something that slows down the disease. First, though, let me remind you of what I'm reminded constantly about, which is these Alzheimer trials failed time and time again. How could you possibly imagine amyloid lowering ever being successful? So here there's a variety of agents here. I think there's a dozen or so. Target and mechanism are listed in the middle column. And then their liabilities that precluded them from adequate trial effects are shown on the right. And you can see time and again that most of these where companies and their academic collaborators spent a lot of time, effort, and huge amounts of money to try to do something about Alzheimer's, failed because they didn't actually do what they were said to do. Target engagement was not there. There was no biomarker evidence that they actually lowered amyloid or lowered tau or whatever. So this is really the reason, and so it's not correct to say, well, amyloid trials failed time and again for decades. Not actually scientifically correct. As soon as a trial occurred, and this happened to be aducanumab from Biogen, that actually lowered amyloid, one right away could see that there was a relationship between amyloid lowering and some slowing of cognitive decline. So the immunotherapy came from Dale Schenk's original experiment in 1999, where he injected APP transgenic mice with saline on the top or A-beta peptides on the bottom, and found this dramatic antibody-mediated removal of A-beta, which began the idea of immunotherapy. And then we know that that went right into an active vaccine that didn't work out at all, produced side effects of a T-cell epitope. And eventually, other antibodies came along, such as Biogen's aducanumab that they licensed. And here, very striking evidence in a one-year trial, their prime phase 1 trial, that on the left was the baseline amyloid PET image with red and yellow, indicating too much amyloid. On the right was the one-year result. Placebo didn't change, of course, but there was a dose-dependent decrease. So this was very striking phase 1 data, and then all the Sturm und Drang around the aducanumab development program that I think everybody here knows well, eventually leading to evidence that one of the two trials seemed to slow cognitive decline, but another one didn't. And that brings us all the way to the headline that I started the talk with, lecanemab in early Alzheimer's disease, which trial done by Eisai and its collaborator. Biogen was a collaborator in this, but Eisai did all the, I think, intellectual and medical lifting, led to a number of outcomes that you see here. And that is on 5 different outcome measures. Lecanemab hit its endpoints in each case. Over 18 months, there was a dramatic reduction in amyloid levels, in yellow here, compared to placebo in blue. This was the primary endpoint, CDR sum of boxes on the top, a small but significant 27% less decline, and 8S-COG, ADCOMS, another cognitive measure, and ADL, which I mentioned, activity of living, is what my patients really are most concerned about. Notice that over 18 months, these trajectories by and large widen. That is, the clinical outcome measures get a little bit more robust as time goes on. And I won't show you today data that was presented at the AIC in August showing that if we take patients who are very mild but do have clinical Alzheimer's disease, those patients do remarkably well. Some patients have a leveling off of decline over 36 months. This is work that Chris Van Dyke presented. So taken together, as the FDA did, if you make a trajectory between amyloid PET on the x-axis, lower amyloid PET to the left, and CDR sum of boxes, and lower is better here, you have a number of trials, that many of which didn't work, some of which did, that lead to the lower left corner. That is, more amyloid reduction over 18 months leads to less cognitive decline. And so as of today, amyloid and A-beta oligomers facilitate tau alteration and tau spread and stimulate microgliosis. Lowering amyloid can lead to lowered tau, and this is a very important point because tau is an independent indicator of neuronal dysfunction, and its levels go down when you target amyloid. So far, there hasn't been great success targeting tau directly, but this is something we want to do to combine treatments. So A-beta antibodies modify both defining lesions of Alzheimer's disease. Amyloid lowering in other human amyloidoses has produced clear-cut clinical benefit, and a great example of that is transthyretin, with 3 FDA-approved agents that change the degree of heart failure from transthyretin amyloidosis in the myocardium. So many factors contribute to Alzheimer's pathogenesis, and A-beta has emerged as the most scientifically compelling therapeutic target and has now been validated in the clinic. And my last slide just makes the point that there are unsolved questions on the road to initial success. Can anti-amyloid doses be lowered by at least 50% or stopped after 2 years, as Eli Lilly is saying with its prescribing information for donanemab? Will combination treatment be substantially more efficacious, such as an A-beta lowering agent plus a GSM, a gamma-secretase modulator, a mechanism that I think is very important, tau targeting with ASOs, as Biogen is doing now, inflammation and oxidative injury. So I think those combination therapies are potentially exciting. And will active A-beta vaccination be safe, efficacious, and less costly? We'll see in the future. Thank you.